What happened? Maybe we should look into it.
Now that Mario has ingested the shiitake mushroom, Lentinan is now in his system. The following events take place in the body in order to stimulate a response in his immune system. Shiitake mushroom is a part of the group Basidiomycota, and while this mushroom contains various compounds nutrients that may support his immunity, heart health, and daily dose of vitamin D, shiitake contains lentinan, which could cause a reaction in 2% of people in the skin's dermis when shiitake mushroom is consumed raw. Lentinan is a toxic polysaccharide that activates IL-1 secretion. Symptoms of lentinan in the body include a rash, swelling, fever, diarrhea, and tingling limbs, hands, and feet. When lentinan first enters the body, it goes to the first line of defense where the innate immunity takes place. Innate immunity is the body's first defense externally and internally against a pathogen through barriers, cells, cytokines, and complement systems. Externally, the mucous membrane within the tract aim to catch the invader, lentinan, and have secretions such as defensins and lysozyme that tear up the invading pathogen when ingested. In addition, cilia within the mucous membrane aim to move the invader into the stomach if defensins and lysozymes fail to break down the invader. Cilia push the invader into the stomach where gastric secretions can destroy the fungi due to the low pH. These mechanisms allow for protection when lentinan did not enter the bloodstream. When it has entered the bloodstream, the innate immune system goes through an internal mechanism. We begin with cellular defenses, where phagocytes play a major role in the destruction of the invader. Phagocytes engulf the invader and destruct the contents in order to take parts that benefit us, such as peptides and amino acids, and remove the toxins. Macrophages are also used to engulf and destruct. This begins with monocytes that go to the damaged tissue and become macrophage, then present to antigen-presenting cells as non-self recognition check. Natural killer cells are also a major component of cellular defenses, where they target and destroy those through perforins and granzymes. Perforins put holes in the invader, which cause a concentration difference and make the invader swell and burst. Granzymes enter in the invader and trigger to make the cell undergo apoptosis. Chemical defenses include complement proteins and cytokines. Complement proteins begin with the C3 convertase generated, which cleaves C3 protein into C3A and C3B fragments. C3b binds to the surface, marking for destruction by phagocytes. In some cases, the cascade accumulates in the formation of a membrane attacks complex, which directly disrupts the pathogen cell membrane, causing lysis. In addition to the chemical defenses, cytokines play a major role in which interleukin and interferon call for T cells, which will be discussed later in the adaptive immune system. Cytokines are responsible for many jobs in the immune system, but a common trend they have is allowing for the gradient where immune cells follow in order to help facilitate repair. Finally, physiological responses include inflammation and fever. These occur due to lentinan infecting the cell, which is attached to vasculature. When the cell is exposed to the toxin, it is recognized as damage, which releases histamine, which promotes a pro-inflammatory response. This causes vasodilation in the vasculature system, which brings more blood to the infected area. Vasculature permeability is also increased, which allows for redness, heat, pain, swelling, and consistent symptoms of lentinan toxin infection. All of these are the first-line defenses that are known to kill the majority of invaders, but if not, the body will undergo adaptive immunity.
Adaptive immunity provides specific defense and memory against invaders by activating T and B lymphocytes. Lentinan is recognized by innate immune receptors like Dectin-1, prompting dendritic cells and macrophages to process it, present peptides via MHC, and release cytokines to recruit immune cells and trigger inflammation. Once cells form and mature in the bone marrow, naive B cells move through the lymphatic system and continue throughout the body. At this point, naive B cells encounter lentinan, which activates the maturation process for B cells. B cells themselves have antigen-specific receptors that code for the DNA within lentinan. When the naive B cells encounter the lentinan antigen and match with the membrane-bound antibody, the cells divide to either become a memory B cell or an effector cell, or in other words, a plasma cell. This allows for antibodies to directly bind to the site. Lentinan itself has to effectively bind to the naive B cell site to induce differentiation. The memory B cells express the same membrane antibody as the original B cell. Antigen-independent and antigen-dependent processes help detect if the Lentinan antibody will continue throughout the body or apoptose. The cell can go through one or two ways, with the ending result being either a memory or a plasma cell. Plasma cells secrete antibodies that work inside the body to free pathogens that are circulating. When the naive B cell divides and further differentiates, the plasma and memory B cells are made. On the other hand, T cells once formed in the bone marrow are now known as progenitor cells that migrate to the thymus in order to start their maturation process and become mature T cells. Developing T cells start to express TCRs and CD4 as well as CD8 receptors. T cells, though, only select for one or the other, so some T cells will select for CD4 while others select for CD8. T cell receptors do not bind to the antigen directly, rather they induce a major histocompatibility complex, either in class 1 or class 2 molecules. These specifically are membrane-bound and are antigen-presenting on cells like dendritic cells or macrophages. T cells go through a process of rearrangement, causing an infinite amount of recombination of genes expressing that specific reception. This allows for a lot of binding diversity, but can lead to attacks against itself cells and molecules. When Lentinan attaches to self molecules, allergic reactions take place. Although T cells do not directly impact Lentinan in the body, Lentinan acts as a modulator for T cell activity. The cytokines in the innate immune system activate T cells. In order to ensure that T cells perform the correct function once matured, they undergo two selection processes, positive and negative selection. Positive selection makes sure that class 1 and class 2 molecules are able to distinguish between self and non-self proteins. Cells in this selection process must be able to bind only to self-MHC molecules, but in the case that these cells do not pass the selection process, they are taken out through apoptosis. Negative selection is for the ability of the cell to be self-tolerant. Moreover, this type of selection codes for the binding ability for CD4 and CD8. For this case, T cells will only bind to the MHC molecule that presents Lentinan. If this fails, negative selection will be eliminated in apoptose. After positive and negative selection occur, three types of mature T cells are left: helper T cells, cytotoxic T cells, and regulatory T cells. Helper T cells express CD4 and aid in the activation of T cells, B cells, and other immune cells. Cytotoxic cells express CD8 and help remove Lentinan infecting host cells. And T regulatory cells express CD4 and other receptors. T helper cells would activate the immune system by signaling to other immune cells like cytotoxic T cells, while on the other hand, cytotoxic T cells would directly destroy any cells presenting Lentinan as a foreign antigen, and regulatory T cells would act to weaken the immune response to prevent excessive inflammation, ensuring that the body doesn't overreact to the Lentinan. Once activated by T helper cells, cytotoxic T cells directly kill cells that present Lentinan as a foreign antigen and on their surface by releasing cytotoxic molecules like perforin and granzymes, leading to cell destruction.